NeoBay Robotics Forum is an academic platform launched by Science Robotics in collaboration with the Institute of Medical Robotics, Shanghai Jiao Tong University, aiming to promote the latest advances in robotics and allied technologies, as well as ongoing basic science research and engineering challenges. The online forum is open to the general public and held monthly with speakers from leading research organizations and industries around the world.
Try to give you an underview of the science that's the basis for a lot of this work. So that's what I'm going to try to emphasize today. So I'll start with just a quick movie to give you an idea of what we were able to do.
4 years ago, we visited his lab, and Schwartz showed us how he implanted tiny sensors like this one into the brains of monkeys, and then wired them to a crude robotic arm. Schwartz told us that when the monkey thinks about moving his own arm, his brain cells or neurons fire off electrical signals. The sensor in his brain can pick up these signals and send them to the robot.
And it's just sort of a chintzy cartoon, but it gives you the idea. What you would see is a lot of activity, not only in motor cortex, but actually through the entire brain. But the pattern would be, you know, there's no obvious pattern here if you would look at these neurons as they fired, okay? And so the real problem from an engineering point of view is how do you extract information from a set of data that look like this? What we started with was we had a monkey. The monkey was looking at a virtual reality monitor. He was moving his arm in a virtual, basically oval that was floating in front of him. And we were tracking his hand and moving a cursor that represented his hand so that he would see this shape in front of him. He'd put his hand in it and he'd move it around a bunch of different times. And so, you know, here's a movie. This is now more than 30 years old. And you're going to see a monkey moving his arm in free space. We're tracking his arm with this marker right here. And we're recording a single unit from his motor cortex. These are these action potentials that I was talking about. They're relatively large. They're easy to see if you can isolate them properly. And what we're interested in is how fast this neuron fires while he moves. And so you can actually look at the relationship between the way the neuron fires and the way the monkey moves its arm. So here you can see it. What we did was this simple algorithm called the population vector. And so we took those preferred direction vectors, those B vectors, and we took one for each of the many cells that were recorded. In this case, there are about 300 cells. And we put a vector down in the preferred direction of each of these cells. And we just adjusted the length of the vector by how fast the neuron was firing. And so we took snapshots of each neuron's firing during a movement. And then we just added these vectors together for each instant of the movement. And we get this yellow vector. And this vector was what we call the population vector, right? So this is just a way of adding the contributions of all these vectors together and getting a resultant vector that represents our decoded movement. And so we can do this again on an instant by instant basis and link these vectors together. And so these are real data from real monkeys that were moving in virtual reality. Okay, and you can see that the monkey was drawing a spiral from outside in and inside out. So, okay, and so again, to review what we're doing, we take these microelectrode arrays, we put them in the motor cortex of these muscles, of these monkeys, record action potentials from individual neurons. We find the preferred direction of each individual neuron. We add these all together in these clusters to make a population vector. The population vector gives us a velocity, instantaneous velocity. We feed that into a robot controller, and the robot controller changes that into velocity at the endpoint of the robot and moves this on an instant by instant basis. So this is the experiment that we did with a monkey. As we said, okay, that was good for 4 degrees of freedom. Now we wanted to add a wrist that was mobile. So we added a wrist that has 3 degrees of freedom, and then we had an animal reach to targets that we had to orient and grasp them. So we put them again in arbitrary places of space. He had to reach and grasp with a robot hand. Okay, so he was successful at that. And again, we expanded our equation now. So now he's reaching in free space. There's yaw, pitch, and roll of the wrist, and he can close the aperture. So we're now up to 7 degrees of freedom. Okay, so now we said, well, okay, what about fingers on the hand? And so what we did is we presented a wide range of objects to the subject, and he had to use his own hand to grasp these objects. We tracked his hand with a 12 camera system, so we could get 22 degrees of freedom and get all the joints of the finger. We decomposed those into principal components. So we had 4 principal components describing the hand shape. We added that to our equation. So now we have reaching in free space, wrist movement, and now we have these 4 dimensions of hand shape. And here's the animal doing this, so we could decode this. And you can see his actual hand movement as he grasps these objects and what we're able to decode. Another thing that we need to make this more robust is we need sensory feedback. So in our second subject, Nathan, we implanted electrodes in the sensory cortex. So this is the motor central sulcus. Here are the 2 that we implanted in the motor cortex. We implanted 2 arrays in sensory cortex so we could stimulate using the sensors in the fingertip so that he could get some feedback of object contact with the sensors in the robot finger. Here he is doing this. This is Rob Gott, the investigator that led this. And what he is doing is pushing on the fingers of the APL hand. And Nathan's able to reply. Index. Right, so we're stimulating the appropriate place in sensory cortex, and he's able to recognize which finger is making contact.
So my talk is going to focus more on the human application, and also instead of using invasive approach, and we focus on developing non-invasive approach to map the brain dynamics and then to accomplish brain-computer interface or brain control using non-invasive EEG recordings. So this cartoon illustrates my talk. On the left side, I'm going to start with the brain mapping first, because that is the scientific basics and highly relevant to some of the work I'm going to present about the human brain-computer interface. The second part of, or the majority of my time, is going to talk about how we could use non-invasive EEG recording, and instead of from some selected neurons, but from large number of neurons, and to decode the intention and control a device. This is a little bit of basic slide just to illustrate and connect the non-invasive EEG and MEG recording with the neuronal membrane excitation. So there's an entire process. You have transmembrane current flow through the passive volume conductor, generate the voltage drop, which can be measured using electrode, which is our EEG, or the current is going to generate an external magnetic field. So if you would use a coil, you're going to record an MEG. Now, the next question would be, how could we be sure? Because we are not going to put electrode inside the brain to record in healthy human brain. And how could we be sure the imaging results is correct? So we have studied this to validate the technology which we've been developed, and in epilepsy patient, because in epilepsy patient, which is there's a particular clinical need, you could put an electrode and over the brain surface or inside the brain to record intracranial EEG, and for the purpose to localize the seizure generating and the region. And so we use that clinical and means to validate our non-invasive EEG source imaging technique. I'm not going to talk about the imaging algorithm. Essentially, it started with a surface EEG recording. We obtained, in this case, it is something called interictal spike, which is a typical biomarker about epilepsy without seizure occurrence. And here is illustrating our non-invasive source imaging result. This color map illustrating where their neuronal and the sources corresponding to this interictal spike. And on the right column illustrated is clinical finding. So this slide illustrates a little bit, particularly focusing on a non-invasive and a BCI, but certainly if you put the electrode inside the brain, which is applied everything else. For example, this is the monkey work you already saw. So with this recording and the brain-computer interface, and one is a notion is a brain decoding base. It's from brain to control a device. You record, you decode, extract a feature, and translate the feature to control command to replace, restore, enhance, supplement, or improve the motor function. But if you want to assist the patient, you want to be something which is more of help. So we say, can we actually control a robotic arm using EEG? In this early experiment, what did we do is we using just a surface EEG, and then we decode and control, and the subject you can see can grab it's a slow, and this is the one limitation of the non-invasive BCI. That is because the EEG signal has low SNR, so it's relatively the control speed is low, and which is not as precise as invasive and BCI, which you're already seeing and in Andy's talk. But on that hand, this can be essentially applied to virtually everyone. So in this particular work, we tested in 12 human subjects, and they all on average performed 80% accuracy in the goal of grabbing and placing the Lego and in this experiment. Now, the concept we are proposing in the field of non-invasive and EEG-based BCI is to use this EEG source imaging. So in a sense, by using EEG source imaging, instead of we doing the BCI control from sensor EEG recording, which is usually a sensor EEG-based BCI, we do conductor model and do source current density and the source imaging, and then we do close this loop. So this is the online system just implement what we discussed. And then we conducted an experiment. This was published in Science Robotics. In this work, we studied in total 68 human subjects. So this is one of the unique point of the non-invasive BCI is we could study a large number of human subjects, and we can train and apply this technology in most of these human subjects. In this particular work, we look into two aspects. One is so-called user learning. Another is machine learning. Machine learning involved essentially is performing source imaging. So the machine and actually they do some signal processing machine learning in the computer side. Now in the user learning, we look into two training paradigms. One is so-called discrete and task. That is sometimes also is called center out. You know, if you have a cursor center out, you could left or right, up or down, that will be, we consider discrete and trial. Another is so-called continuous pursuit. That is, if the cursor is continuously moving randomly, and we are training the subject to follow that randomly moving and the trace of the object. So we conducted a number of training and control experiments. Finally, we tested our technology in a group of 6 human subjects to actually controlling a robotic arm.
My talk will rather focus on the future and will show what we can learn from neuroscience for future ways how we build robotic architectures, in particular new algorithms for robots. So this is just a couple examples. In neuroscience literature, we can find a lot of knowledge about different parts of the brain, different circuits in the brain that seem to perform different functions. So what can we learn from biology? On the one hand side, we could learn specific neural circuits for different tasks relevant in robotics. And the example could be different sensing tasks, like for instance, this lumen detector could be used for obstacle avoidance system on a drone, or famous divergence-based landing of drones shown by Guido de Kroon that is inspired by insect circuits. We could also mimic some circuit from the navigation system, so those hippocampal circuits that has been done by Michael Meaford a long time ago in Red Slum, and a couple of groups have recently also replicated that work, developing SLAM architectures that are based on these neuronal circuits and architectures, or central pattern generators that can be used to control locomotion in, for instance, lizard-like robots of Auka iceberg. On the other hand, we could also learn some architectural principles. The first principle states how states and variables are represented in the brain. And here, so luckily, Andrew has already in detail has shown you how neurons represent different behavioral variables. So I also like this example with reaching task for the monkey. When monkey reaches for different targets, and one can observe activity of different neurons in its brain, and one can see that activity of neurons correlates with different direction of movement. Meaning the neurons seem to sense different directions of movement, and different neurons will will kind of react to different directions more or less. Typically, they would react most for their favorite direction, and then slightly less for some other directions. And if you want to estimate the current state of the brain of this monkey in order to guess where it wants to move its arm, you have to look at the population of neurons. You don't look at a single neuron. So this population encoding seems to be an important principle of how variables are encoded, at least in cortex of some animals. So this is just a little cartoon, how that's done. So if you have some parameter of the environment, of the behavior, the speed of movement of your robotic arm, then the neurons would usually represent or sense different values of this parameter with so-called receptive field. So they will have different sensitivity to different values of the parameter. So that each neuron will have some sensation, some reaction to presence of this parameter, and other neurons will have most likely different reaction to the same parameter, so that if you look at the whole population, you can make an estimation. So here, if our neuronal state, the blue curve, forms the representation and makes a decision for a certain value of the variable, it can stay active even if the initial input has gone away. And it will kind of keep this state as long as needed to perform some action, even if the sensory input was occluded or disappeared. So we have these memory properties. These properties can be handy for kind of real-world applications. For instance, if you want to track one of the objects, but there are several objects that have the same saliency, they're basically the same objects, this winner-take-all dynamics can allow us to select one and only focus on that one, ignore the distractors. It can also help us in tracking objects if our tracker is of bad quality. Like here we can see we have a very simple tracker, it's a simple template matching. And if we just try to track the fish with this tracker, it will jump around because it's like really not very high quality tracker. However, if we add these kind of recurrent dynamics and attractor dynamics, it will try to stick to the previous decision of our tracker and will clean out the noise. So the final and important principle is the following. So those attractors, we have created them by creating lateral recurrent connections, and those connections create loops. Create loops between activity of different neurons. And such loops are pervasive in biological neuronal systems and in the brain. And you can find these loops on different levels of description of the brain. As some of you might know that the cortex, so that part of the brain that makes us smart consists of 6 distinctive layers. They have different types of neurons with different type of connectivity. And there's some lateral connectivity that creates loops between these layers, but even more importantly, there are connectivity loops between the cortical layers and the external subcortical structures such as thalamus, basal ganglia, or cerebellum. And these loops, according to some very new theories of understanding the brain dynamics, and we can show how parsing action sequences can be done with this framework, how we can learn action sequences with reinforcement learning, how we can plan action sequences, or how we can learn different forward or inverse mappings autonomously by activating and deactivating learning processes as needed. These processing loops are not only used in order to predict something in the future and then maybe check whether this prediction has taken place. They also can be used to check consistency at this moment in time. And this consistency can be checked for perceptual tasks. So if we want to perceive some object and we know that this object has different aspects, has different features that must be present at the same time, then we can create kind of loop-like structures, which I won't go into detail here, that allows us to check are all the attributes of the object that I know belong together, like apple is red and round and smells like apple, that they are all present and non-contradicting aspects like long for apple are present. However, the new and emerging type of computing hardware, so-called neuromorphic hardware, fits the structure very well. So neuromorphic hardware typically would support parallel sparse compute, asynchronous and event-based computing, which again allows the system to activate and deactivate modules and enable communication between them on demand, making for very efficient implementations. And the systems usually support learning on the fly, so that the system can reconfigure itself and be plastic and adaptive on the fly. It has been shown that these neuromorphic chips, neuromorphic hardware, is particularly advantageous for recurrent networks. So for the type of the networks that I have been talking about with all those recurrent loops. There's a big question on how to program these devices, and then there's no trivial answer. So we at Intel are developing a novel computing framework and software framework, Lava, that will allow us to configure these networks with very rich kind of structural motifs that can be used for different parts of the architecture and for different tasks.